Okay, let me start once again thanking the organizers and also our chair, Kunjan. Thank you. Any questions on the last two lectures? Okay. So as I announced last time, yesterday, the next topic is going to be on dynamics of charged macromolecules. This is a difficult topic for obvious reasons. Already we talked about conformational entropy, long-range correlations of them. And then we talked about electrostatic interaction, long-range correlations. That's in equilibrium. Now, we are trying to learn a little bit about how these molecules are going to move from one place to another place. How are they going to do that? And in nature, there are many driving forces. There's a concentration gradient of polymers, concentration gradient of salt ions, and then different traps to absorb them, to capture them. And there are several places where they will release them. So there is really a highly orchestrated process. But fundamentally, how these molecules are going to move around are going to be important. If they are going to be static in place, then we would not have life. They have to move, right? The way I think, the way I communicate, by this time, many molecules have moved. And lots of ions that have controlled their movements. This is an extremely complicated problem. One lecture would not be enough to go into the details of how these processes would take place. So I'm going to scale back. And again, like what I said in the last two lectures, let's take a baby step. Let's do the simplest thing. And already we'll see some challenges to understand. And that's the purpose of today's lecture, to share with you what the complexity is, how one could possibly go and try to figure out how this is happening. Then based upon such a ground floor foundation, maybe we can put additional details and go forward in terms of more realistic problems. So even there are lots of issues, right? In terms of viscoelasticity, mechanical properties, elasticity, et cetera, right? And mobility, how a molecule is going to be drifting from one place to another place, charged molecule is going to drift from one place to another place. A lot of things are going to happen. But in the interest of time, and also for the sake of being clear enough so that you could all participate in the discussion, I'm going to focus only on diffusion, right? And I'm going to take a two-sided approach to this diffusion. One is going to be the simplest situation of only a few molecules in solution, dilute solution or maybe semi-dilute solution. Only a few molecules are hanging around, charged molecules are hanging around. The other extreme would be, I'll go to the other end. Well, I like to talk about a crowded environment. And in the middle, there are so many interesting things that are happening, which we'll not talk about because of the interest of time, right? These are the two limits that I like to talk about. So today's focus is going to be on diffusion. So let me go and share with you, if this would allow me to do it. Oh, thank you. So what do we have? I have been telling you this already twice. So I have a large macromolecule, very large, charged. In this particular case, you can think of a single-stranded DNA, RNA. And this is placed inside a bulk, salty water. We have lots of salt ions, salty water. And I have mentioned that the function of this molecule, structure, mobility, dynamics, internal dynamics, all these things are arising from combination of topological correlation due to chain connectivity, electrostatic interactions because of charges, and also hydrodynamics. And water, as we can easily acknowledge, that's the matrix of life. Without water, we are nothing, right? We won't, this planet is not suitable, right? And how do we think about water? Well, immediately, we'll think about the hydrogen bond, Let me think in a bigger scale, right? Ranjini, first time we met at a conference, we were in Trieste, and this is the picture of Trieste. It is indeed. Okay. No, this is more recent, but that's a place, right? Anyway, so we were at, the institute at Trieste. That's how we went around. We went to Venice together, several of us, right? Anyway, so look at this. Suppose I have a sailboat here, right? And I will make a wave. On the sailboat there would feel the effect of the wave that I'm making and vice versa. And how do I describe this? How do I describe the effect of whatever perturbation I'm making in here? Suppose I put a force in here, what will happen in here? And then that is going to create certain things. Another sailboat there is going to create another wave. There's going to be a tremendous amount of orchestra here. Right? How do we think about this? I want to share with you the simplest way we think about these problems. And since the time scale is kind of long, we're not talking about very short microsecond or nanosecond time scale. We're talking about very long time scales. For that, we could write the linearized Navier-Stokes equation. Eta naught del square V at some location R plus a grad P pressure at that location R. That's equal to any force that we write. We can impose gravity, electrical force, whatever force we can put, the force at the location R. And you, I'm sure, have seen the solution of this equation. The solution is, suppose this is incompressible. Water is practically incompressible liquid. Under those circumstances, namely, incompressible means del dot V is equal to 0. This gives me a solution of this equation easily. V, velocity at some location R, is equal to dr prime, and there is a tensor R minus R prime dot F at R prime. What this means is this: in this ocean liquid, if I were to put a force here, some location here, there is some force, a location R prime. Far away, I mean, I look at a distance at R, a position at R, and velocity at that location R is dependent on the force in here. So there is a hydrodynamic perturbation, a wave that is connecting force to give me the velocity here. How do I write this? And that's given by here. This is, as you know, have you heard about this? Wolf's tensor. And let me write it down explicitly what that is. G at a separation distance of R, that's equal to 1 divided by 8 pi viscosity of the liquid, namely water in this particular case. Let me write down 1 over R. And the tensorial part, vector R by R squared. Okay. And right away you see this character, 1 over R. Remember topological correlation, density profile dependent upon R like 1 over R, monomer density from center of mass. And we know the electrostatic interactions 1 over R. Now hydrodynamic interaction is also 1 over R between 2 points. This is also long range. So in real world, when you talk about these Coulomb systems, we are talking about 3 long range interactions. And clearly, in addition to the double screening that I talked about yesterday, we need to talk about triple screening because there are going to be molecules coming in here. This 1 over R is going to be screened. So there is going to be triple screening, and we have to do it self-consistently, and they're all operating simultaneously. Okay, so now what I have is, my challenge is when I put an RNA or DNA there, so I suppose I have a polymer chain in here. And here is my ith segment. And so let's say this is my jth segment. So the jth segment, because of friction against, this is going to be in water, this is going to create some force. That force will travel through this fluid and affect the velocity of this monomer. And this monomer itself will have a friction against the force that's created or velocity that's created, right? Friction times velocity will give me a force. So this will emanate a force. That force will go and affect that one, will affect this one, and this one will affect that one, and this one will affect this one. So we have enormous amount of correlations among various monomers, hydrodynamic interaction. This is really amazing. So this is an incredibly complicated situation, right? There is a huge orchestra. One monomer rings a bell, another monomer hears it, another one rings a bell, another one rings a bell. There's a lot of ringing bells at the same time, and lots of correlations, and then we have beautiful music. Right? So that's what we are going to do. How are we going to calculate this? How are we going to address this? Next one, please. Next slide. Okay, this slide is exactly what I said here, right? So in general, I'll write the Newton's second law of dynamics. So that's the typical nonlinear, the inertia term, nonlinear term. Today, I'm not interested in turbulent flows. I'm interested in very simple systems. So therefore, under those circumstances, I don't have to worry about nonlinear terms. And for water, you should know the viscosity is 1 centipoise. The mass density is 1 gram per cc. That's my water at room temperature. At a low frequency, long time scales, I linearize this, and then I just have written exactly what I said over there. This is very difficult to deal technically, right? How do I put this in and then try to figure out what the diffusion coefficient of this molecule is? How would I do that, right? That seems to be an ambitious task, right? Because of the complexity I'm telling you. However, there are boundary conditions in thinking process. Whenever we think about something, there are boundary conditions. What are those boundary conditions? These are beacons of light. It's the great people who had worked on it, right? And one such thing is a Brownian motion of simple particles. So let me consider a Brownian particle of mass m, the radius is r, and let's say it has a friction coefficient against the background. Let's say viscosity is eta naught, or let me say eta naught, right? That's viscosity of the liquid, namely in this particular water. And I like to write the dynamics of center of mass of this particular time t. With respect to some coordinate system, that's the center of mass at that point. Little later, it's going to be moving around. The dynamics of the center of mass would be second derivative of this with respect to time, acceleration, mass. That's equal to minus some friction coefficient zeta times dr center of mass at time t by dt, that's the velocity. And then there is some random force. Water molecules are bombarding against this particle. And then I have some random force acting on that particle. And this is a Langevin equation. Then you know that the fluctuation dissipation theorem connects the correlation function of this F with respect to friction, etc. But nevertheless, at the end of the day, for long enough times, for this particular problem, for long enough times, which is larger than m over zeta, which you could do by inertial analysis, for such conditions, the mean square displacement, center of mass at time t, minus center of mass at 0 time square. This is the famous diffusion equation, 6 times dt in 3 dimensions. D is the diffusion coefficient of the particle, spherical particle. Now, what is D? This is Einstein. D is temperature dependent. KBT, T is temperature in absolute degrees Kelvin, divided by the friction coefficient. So D is never 0 for finite temperatures, right? If somebody like me says D is 0 at room temperature, all right, you have to check me out, right? D cannot be 0 at finite temperatures. Okay, now, even 50 years earlier, 60 years earlier than Einstein's work, Stokes showed the friction coefficient of the sphere against the fluid is equal to 6 pi eta naught times the radius of this sphere. Radius of this sphere. That's Stokes. I found this to be remarkable. When I was younger, when I was trying to learn about this hydrodynamic problem, Stokes problem, I derived Stokes problem, and I could not, because I did not believe it. I did not believe in a young person, we don't believe everything we hear, right? Hopefully. So why was it not intuitively obvious to me? So here is my sphere, right? And this is undergoing friction in the fluid. I would have thought the friction coefficient should depend upon the surface area, because the surface area is the one that is exposed to water. So there is a friction everywhere. That's what I thought. So immediately when people told me about Stokes, no, wait a second, I want to do this calculation again, right? Because it's not obvious to me. You do it, that answer is the exact answer. Okay, that's already a breakthrough of the thinking by Stokes. That's true. So now what we do is we combine them. And this is called a Stokes-Einstein. We call it law. Take this, put it back there. That gives me D is equal to KBT 6 pi eta naught R. This is really fantastic, right? If you could measure D, you know the size of the particle. Or if you know the size of the particle, you can tell what the diffusion coefficient is going to be. So that's the structure of the theory. Okay, so in addition to writing this Langevin equation this way and calculating mean square displacement, you could also do a continuum representation of the dynamics of this particle. That is, suppose I write the probability of finding the density, probability of finding the particle in a solution at location R, say at time t, d rho by dt, that's equal to d del square rho RT. This is equivalent to this equation. You have seen this like Fick's law, for example, simple diffusion equation. And this is also Smolhovsky or Fokker-Planck. There are many versions of this, depending upon which book you follow for theoretical physics. Okay, let me do a Fourier transform this. Then I do this d by dt, rho k is actually scattering wave vector, but the Fourier variable conjugated to this position variable r at time t. This is equal to minus d k square rho k at time t. That's a very simple equation. We can solve this. So when I do this rho k at t, rho say minus k at 0 time, right? And take the average of this, that will turn out to be, apart from normalization, that's going to be e to the minus d k square t. Right, let me normalize this. Rho k at t square. So the way experimentalists would determine diffusion coefficient in general is as follows. Either you do particle tracking, right? You put a fluorescent label and watch it under the microscope and monitor how it goes. If the particle is big enough, even with the optical microscope, like a Perron did long time back, right? And then you track the molecule, the movement of this as a function of time. Make sure the diffusive law is obeyed by maintaining this condition. This condition is, otherwise you may not see this law. Or when you do simulations, you want to make sure this condition is met if you want to see this. So then you can get D. The other way you can do is to do scattering measurements. If you do light scattering, for example, these molecules, the molecules they're very interested in, have very good contrast in terms of refractive index with respect to water. And that's the reason why I'm able to see fish in my pond, because the protein, the refractive index of protein is very different from the level of water. Therefore, there's a contrast and I can see it. So light scattering is a very good way to do it. If we do this, right, number one, this is proportional to scattering intensity correlation function. So if diffusion is valid, I would see an exponential law. And this would look like e to the minus some rate times t. It will be exponentially decaying. It is, right? Exponential decay with time. The other aspect is this gamma, the rate, should be quadratic with respect to k squared. Because that should be dk squared. So you cannot make measurement zero angle because you'll damage your your detector. So you get the data like this. So the slope should be diffusion coefficient. That's how you determine diffusion coefficient. Okay, let's go to the next slide, please. Next one. Okay, I said this. Next one. Next slide. Yeah, so it's essential, right? When you make measurements, you want to make sure you make multi-angle measurements. You have to make many, many angles, right? Then you make sure it's linear, then you get the slope, then you get diffusion coefficient. Next one, please. Good. So let's go back to reality now. That's the theoretical argument. So what I said is Einstein's calculations and Stokes, that's the result. Now let me think about a polymer. Let's say the radius of gyration of this polymer is, say, Rg. We know that, right? And we know how this depends on molecular weight, depending upon the coil globule transition, stiffness, etc. There's an exponent in n power nu, right? So what we would expect is we would expect very quickly by borrowing this idea that the diffusion coefficient will be like a temperature divided by R. R is the radius of gyration. Then our radius gyration is proportional to the nu. That's what I would expect. Right? Any questions in here? I'm making a bold comment. Yeah. Not yet. Not yet. Please wait. Wait for the feast. Right. Any other questions? Yeah, there should be a question here because look, I have a polymer here, right? I have a polymer here, coiled up, right? And imagine my hard, colloidal particle or whatever it is, right? Solid object. This is lots of holes in here. There is lots of water molecules in here. Why would Muthu think that I could imagine that this is very much like this one? Because it is not solid, right? It's not only surface alone. There are a lot of things, lots of holes in here, right? That's a natural question to ask, right? So I have to do a calculation. Okay, let me do a calculation to see whether these expectations are right or wrong. Let's do this very simply. Let me take, this is the picture that I have. This is the picture that I have. So let me try to account for this one. You might be thinking this guy is greedy. We can do it, right? So here is my thing. This is my I. This is my J. And there is lots of hydrodynamic coupling. Let me say minus Ji. This is force on my ith monomer. For whatever reasons, net force on the ith monomer is minus sigma I. Now let me sum over all i's. That's the total force acting on all monomers from the fluid from various conditions. Let me call this to be, and let me average it out. Let me call this to be equal to the friction coefficient zeta maybe to change the notation, I don't want to confuse it. Ft, translation friction coefficient, that is zeta, right? Put a negative sign, and center of mass of the polymer chain dot velocity. Okay, that's the definition of my friction coefficient, because I need to know the friction coefficient, then I can put it in the Einstein's formula, then I get an expression for diffusion coefficient. So that's what it is. So let's be qualitative. Let's write down an expression for this one. So minus a sigma I would be equal to minus friction coefficient of one monomer. This is one monomer, and velocity of that monomer. Ri dot is velocity of that monomer. This is friction coefficient. Friction times velocity gives me force, right? So that's the force acting on that monomer. But on top of that, I have forces coming from here, and from here. So that means I have sigma j, right? That's a force. I already wrote down the expression for the velocity from the Voisin tensor right here. There is a force being converted by this magical key to my velocity after a certain distance. So what I have then is this force is transmitted to a position at Ri from Rj. So that's a tensor. And summation over j not equal to I, of course. They cannot self correlate. Then I have the force converted to velocity. I'm writing an expression of force. So the velocity will be converted into a force by writing the friction coefficient for that ith monomer, the beads, JB, zeta B. Now let me average it out. This is what is GRIJ. GRIJ is 1 over 8 pi eta naught, distance between the two monomers, two segments, and then the tensorial part. And then it's also dotted with the force on the jth monomer. This is not possible to do exactly. You have to do numerically. But what people did is to make what is called pre-averaging approximation. That means you split this product into the angular average of the product, the ensemble average of the product to be product of averages. If you do that approximation now, minus zeta B, zeta B, Ri dot plus zeta B, summation over j not equal to I, then I have G Ri minus Rj average times sigma j. This is easy to do because that's equal to 1 over, if you do this average, 6 pi eta naught 1 over Ri minus Rj average, this quantity. We know the statistics, the probability that the two monomers are going to be separated. We did calculation of a Gaussian chain. Similarly, for any conditions, we can calculate the probability for this of realizing certain separation of distance between segments. So we can perform this average, right, and substitute that in here. And then what we have is we have a coupled equation, right? We have sigma I is related to this. This is a diagonalization of matrix. When you do this, what do you get? You can calculate this with the paper and pencil with this approximation. If you do all these things, the friction coefficient that Ft turns out to be like this. Ft is equal to, if there is no hydrodynamics at all, it's simply zeta B for every monomer times the number of monomers. This is no hydrodynamic interaction. In polymer literature, this is called Rouse dynamics. Okay, that's dumb. That every monomer is undergoing friction, the total friction is equal to total number of monomers times friction coefficient of each one. In the other situation, what happens is this is the number 6 pi eta naught Rg. That's what happens. It is indeed proportional to Rg. but it's not 6 pi eta naught Rg except for this number. So what normally people do is they write to be 6 pi eta naught times RH, which is hydrodynamic radius, they call it. But the important thing is Einstein's law is applicable even for such a complicated fractal object, because you can do these calculations explicitly, and that's what turns out to be the case. So my expectation of implementing Einstein's law is okay. Okay, so this means, okay, this is with hydrodynamic interaction, and this is called a ZIM dynamics in polymer language, polymer literature. Okay, when you hear ZIM dynamics, that's what it is. It's equivalent to Einstein for dilute solutions. Okay, please. Sorry. No, if there is no hydrodynamics, there is no ocean at all. So they are all independent. They will turn out to be zero, because when you do the connectivity change, then they will balance out.
So then that left-hand side sum should be zero, right? On the top, sigma I, summation over all I.
See, when you don't have hydrodynamic interaction, you will not have this.
Yeah.
Right?
Yeah.
Then when you add the total force, it will be number of terms that you have. This is for each monomer, right? You'll have the total number of capital N, number of times that term will give me the friction coefficient, because the velocity, the steady state velocity of the center of mass of the chain is same as velocity of each monomer.
Okay. Okay.
In that expression, when you introduce sigma I, so that's the force on the ith particle.
That's the force minus sigma I is force on the ith particle.
Okay.
Sigma I is a force on the fluid, minus sigma I is on the particle.
Okay, that was my question.
Thank you. Well, it's an important question, because people do not explain this in the papers or in even the books. It just, that's okay. Please keep asking questions. All right, so the point that I'm trying to make is this, right? So in principle, depending upon how much hydrodynamics you have, whether you have screening or not, you can have an interpolation. You can go from this full-fledged hydrodynamic interaction result, which is Einstein, and then to no hydrodynamic interaction, which we call Rouse, and then this is a crossover formula which you can derive analytically. Okay, next slide, please. Please. Please go back. Please go back. Thank you. It's a different, I don't know at what level you're asking, it's different by this factor of 3 times root pi by 8. Okay. And the reason is, when we talk about hydrodynamic radius, right, we are taking average of 1 over Ri minus Rj. In radius generation, you do the numerator. You take the average in numerator. So when you do the work out the arithmetics, that's the way it comes. Yeah, it's not changing the size. Size is the same in equilibrium. Well, the way we interpret them is like in a shape. The hydrodynamics is able to capture the shape. Size may be the same, but there's a shape factor. So the ratio between Rg and Rh, as Ranjini knows, is called a shape factor. But we are talking about numerical prefactors here. Because the important thing is how does it depend on molecular weight, right? Both of them depend on molecular weight the same way. Because the RG is proposed to be. It's not, yeah, I don't know why you would say that, right? The difference between 3 root pi by 8 and a hard sphere is because one is this one, the holes that I was talking about really results in only 3 times root pi by 8. That's the only difference it creates. It doesn't change the physics. So when you account for the fact that it's a fractal object, it only changes the numerical factor. So the universality of how the friction coefficient depends on this linear size of the polymer or object is the same. Yeah, please. Well, that's an interesting comment, right? But let me paraphrase it differently. The hydrodynamic interaction, the way I talked about it yesterday, if we have another chain, so I have hydrodynamic interaction, right? Hydrodynamic interaction between this and this. When a chain interpenetrates in here, the hydrodynamic interaction is going to be screened. Right? 1 over R interaction now is going to screen with another screening length, which is hydrodynamic screening length. We have topological screening length, then we have electrostatic screening length, Debye length, and then we have another hydrodynamic screening length. So when you go to higher and higher concentrations where you are going to talk about entangled situation, it is not the hydrodynamics of the liquid, namely the water, that's going to dominate, but then the viscosity of that entangled melt itself will have a viscosity, right? That will be not hydrodynamics, oil dynamics, whatever that may be. Yes, then there will be consequences of those interactions. So here, in this particular case, the chains are not yet entangled. Then we transition into another problem. Okay, so next slide, please. Sorry, go ahead. But we never see for flexible chains, we never see a rod particle. We never see it, it's a coil, right? We remember the exponent nu is not 1, it's a half. So is it good? So I made, that's Einstein's calculation, I copied it, I expected something, and I have some sort of a security blanket by doing this calculation. Yeah, when you do this, yeah, it seems to be like that. Let's go to the laboratory and see whether this has any value. Okay, that's the ultimate judge. When we do this, here is an example. This is a good solvent. The exponent nu for a good solvent is 3/5, 0.6, right? And these are the data. Look at the logarithmic scale, several decades, and that's the diffusion coefficient, again, several decades. Pretty good. This is a polystyrene, no charges yet. Polystyrene toluene, good solvent, that's what you get. This is good. We are on firm ground. We can quibble about prefactors, but we are in, this law is valid. Okay, now let's put charges. Next one, please. What would we expect? Again, going back to your comments. What we would expect is, okay, so D goes like temperature divided by RG. And high salt, yesterday we talked about it. In high salt, right, that's like a good solution, right? Because 4 pi over kappa square is a term. LB times 4 pi over kappa square is a term. So therefore, we would expect a D going like temperature divided by n to the 3/5. That's what we would expect for high salt. A low salt, we would expect a T divided by N, very much the ultimate limit. That's what we would expect. Therefore, we would expect when we have low salt, the chain is going to be expanded. not necessarily rod-like, as we pointed out, but it'll be more like expanded. The radius gyration is big, therefore diffusion coefficient is small. Then when I put salt, it will shrink, radius gyration becomes smaller, diffusion coefficient will be higher. Therefore, I would expect to see, right, when I reduce the salt concentration, D should decrease. Reduce salt concentration, chain expands, diffusion would go down. Based upon everything that I said. Let's go to the next slide, please. It is a surprise. This surprise has been around for many years, 5 decades. These are facts, external facts. These are very carefully made measurements. They took polypeptide, polylysine, monodisperse. It's pure sample. There is no question about whether polydispersity or it has multiple charges or nothing like that. Polylysine, it's a polycation. In sodium bromide solution, high salt, no problem. That's a Stokes-Einstein. High salt behaves very well. When I reduce the salt concentration, this is a logarithmic scale. Let's look at one concentration, the 3 milligram per milliliter of this sample. Let's not worry about the other concentration. So when I lower the salt concentration, what happens is the diffusion coefficient goes up. Exactly the opposite of what we would expect according to Stokes-Einstein. Temperature is the same. That would be our prediction. If you did that, yeah, if we predict that, it should go other way around. Right? But that's not what we see. That's not what we saw. Then it keeps on increasing. And people call this to be ordinary behavior. Why? Every laboratory in the world saw this. Then, after reaching some sort of some strange concentration of low ionic strength, in addition to this behavior, they found another behavior. Simultaneously, the diffusion coefficient is not 0. Diffusion coefficient is small, much smaller than this number. And that they call to be extraordinary behavior. So this phenomenon is called ordinary extraordinary transition in the literature. It is not only for polylysine, this is seen for all kinds of polymers, all kinds of charged polymers. Next slide, please. Here is another example. At low ionic strength. So this is, right, this is a semi-log plot. This is scattering intensity, correlation function. So that's an exponential one. They call this a fast, which is corresponding to ordinary behavior. Then there is another slow behavior, room temperature, everything, right? And they call that to be the extraordinary behavior. When I dump salt into this, these two modes collapse into only one exponential decay. That would be Stokes-Einstein-like. This is a fact. And this is true, this kind of phenomenon is true for all kinds of systems, RNA, anything, polystyrene sulfonate, polyvinyl pyridinium salts, proteins, etc., etc. So this is the enigma. As soon as you put a charge system, everything that I wrote down here appears to break down. Why is that? If you don't understand even this problem, we are not going to make progress in going further, right? So this is a big enigma. How do we do this? Next slide, please. Thank you. I don't know how to use it, but this one. This one, okay, I'll try. Okay, let's look at the fast diffusion coefficient. Look at this example. Don't worry about what polymer that is. This is very generically true. Now, here is already a decade of polymer concentration and different molecular weights of the same sample, same polymer. Look at the diffusion coefficient. It's the same. And if you pay attention to this, that's about 6 times 10 to the minus 6 centimeters square per second. For a small ion, like potassium ion, it's a 2 times 10 to the minus 5. This is only about 4 times smaller than that of a small metallic ion, diffusion coefficient. This is totally bizarre. This is a fact. Next slide. Oh, I'll do that. Okay, now I put all the data together from many laboratories in the world, right? This is the concentration, this is the fast diffusion, and it does not depend upon molecular weight, does not depend upon concentration of the polymer in the solution, it's a salt-free. And then there's the other mode, the extraordinary behavior, that's a slow mode, that will depend upon the polymer concentration as well as the molecular weight. Okay? And we'll come back to this slow mode later on, but this is very fascinating. If large molecules can be pushed to move, diffuse, just like a small metallic ion, this is really fantastic. So our large macromolecules can be moved around by playing with concentration gradients. Let me try this one. And the answer lies on this picture. I told you several times, the molecule is not alone. Einstein's law is for an isolated particle. But in a charged systems, the molecules are not alone, right? The molecules are always surrounded by counterions. Look at that. There is always a cloud hovering around the backbone of the polymer chain. Therefore, if I want to talk about the dynamics of the polymer charged molecule, I better deal with this quasi-particle polymer and its shirt, namely the cloud. I have to really work out the dynamics of them together. Otherwise, I will not be able to explain the experimental data or the way the molecules are moving around the real world, real systems. So that's the idea. So net charge is not equal to the chemical charge. We talked about it. That's because of counter adsorption. We talked about dielectric mismatch, et cetera, et cetera. But the counter ions, they have their own dynamics. We have to do them together. So how do you do this? Let's try to compose an attempt, and then I'll show you the slide. Okay, so I have this equation, right? Let me call C1 to be the polymer concentration, dC1 by dt at some location R at T. That's equal to minus divergence of the flux. And the flux of the first kind, that would be negative diffusion coefficient of the first kind, grad C1 or T. This is what I wrote down so far, like Small-Hofsky equation, right? You take this J1 and put it in here, then you have a plus diffusion coefficient del square C1. That's the Small-Hofsky or Einstein's equation. But now, since I have counter ion, there is also a convective term. Convective term will give me a push to this monomer, the flux, which would be the concentration times velocity. From velocity, that would be flux. Density times velocity would be the flux. How do we get the velocity? The velocity, again, this was done by Einstein. Velocity is like this. Suppose I have friction coefficient here, zeta, for the whole polymer chain, right? And then velocity, that would be equal to the total charge on that object times electric field. Now, this electric field is not created externally. I'm not applying any electric field at all here. The electric field is created with a counter ion cloud. So let me call electric field local. Therefore, the velocity would be Q divided by zeta, E local. And this is called electrophoretic mobility. And this is Einstein's electrophoretic mobility. Einstein's electrophoretic mobility. And that mu is equal to Q by zeta using Stokes, Einstein's law, that's a QD by KT. Okay. So therefore, this term becomes C1 RT mu1 and mu1 times E local. So therefore, when I look at dC1 by dt, that would be equal to plus d1 del square C1, bring this in here, plus now del dot, sorry, negative, del dot C1 RT mu1 E local. And we know del dot E, right? We had it day before yesterday. Del dot E is equal to 1 over epsilon naught, epsilon times the charge density. Charge density is the total charge of the polymer, right, times concentration, right? And then plus here charge of the counter ion, the E times concentration of the counter ions, C2. So therefore, when I write the equation of motion for C1, they run like this. There's a term which goes like C1. There's another term which depends on C2. Similar to this, I'll be able to write an equation of motion for the second species, and the counter ion, dC2 by dt is equal to that. Therefore, I get two equations which are coupled to describe the coupled dynamics between the cloud and the polymer. So that's what this slide is about. So C1RT is the local number concentration of the polymer at that location R. And then the conservation, mass conservation equation gives me this equation, which I just wrote down there. J1 is equal to minus D1 grad C1 plus CRT mobility times local and induced electric field. And then I know the del dot E is this quantity. I know that. Next slide, please. And then I look at some fluctuation because that's what I measured in light scattering experiments. So there's an average value plus delta C1 fluctuation. This fluctuation counter ion cloud concentration. Then that expression turns out to be like this. And then I have one equation like this, another equation for this one. This depends upon delta C2. Delta C2 depends upon delta C1. They are coupled. And this is a 2x2 matrix, trivial mathematical example. Now we can solve this very quickly. Exactly. The next slide, please. The answer, when you do this, the answer is this. Two coupled equations, two eigenvalues. One eigenvalue is not diffusive. It turns out when you do the analysis, it's not diffusive. It's called Plasmon mode or Debye mode. And it's a super fast. You cannot detect it in a light scattering. We still have not managed to make measurements in the world to track that super fast motion because that's a plasma, that's a cloud dynamics. The other mode is a diffusive, and that's a fast diffusion. That answer turns out to be given by this one. Okay. And let me not bore you with the technical details, right? I can substitute, given all those parameters of a problem, the way we describe this, and I look at this quantity for the salt-free case where we are seeing this faster diffusion. The answer turns out to be the coupled diffusion turns out to be the diffusion coefficient, Stokes-Einstein diffusion coefficient of the polymer plus 1 plus this one. This term is coming from the coupling. That term simply is a degree of ionization times the number of monomers, right? And D1 is a monomer diffusion by N, because we know for low salt concentrations, diffusion coefficient goes like 1 over Rg, and that is Rg is proportional to N. So when we put this one here, D1 is 1 over N. This alpha N is much larger than 1. N is 1,000 or something like that, right? So much alpha is like 0.25, 0.3, something like this. So this is much larger than 1. Therefore, this simply turns out to be alpha times the D0. D0 is a number, alpha is simply a number. So basically, D0 is diffusion coefficient like a metallic ion, and the fast diffusion coefficient is simply degree of ionization. And that is of the order of 0.25, 0.3, 0.4, 0.2, something like that. That's the reason why the fast diffusion coefficient is so high. And that's the reason why it does not depend upon molecular weight. I showed you. And also, if you follow the analysis, it also does not depend upon the polymer concentration. So the explanation of this enigmatic faster diffusion that we have been reporting in the world is simply coupling between the counter ion cloud and the polymer. Counter ions, counter ions, counter ions. The polymer, charged polymer is not alone. It's never alone. Now, on the other hand, if I go to high salt, when I go to high salt, kappa square, as we know, is proportional salt concentration. And that's in the denominator. If that's high, this gets killed. The coupling is broken because of screening, the coupling is broken. Then I recover Stokes-Einstein. Everything's okay. And this is a kind of a baby example, given the context. This is the simplest example where I can communicate the importance of the coupling between the counter ion dynamics and the polymer dynamics. This is manifested throughout the systems, throughout every system. Sequential polymer in IDPs, polysaccharides, charged polysaccharides, everywhere you talk about you have this problem. The way you analyze the data is going through this kind of description. Next one, please. Okay, so let me go to the other one. What is this about? I hope you have already a clue in terms of explaining why this could come about. Because I gave you the hint yesterday. Look at this, the diffusion coefficient here is about 2 orders of magnitude smaller than what you would expect from Stokes-Einstein, right? Knowing D goes like 1 over Rg, if the D goes down by 2 orders of magnitude, D goes down by 2 orders of magnitude, that means the R is increasing, right? No salt, right? The chains are supposed to be far away from each other. But still, when you do light scattering, you seem to detect large aggregates. Otherwise, you would not see this. Okay. Next slide, please. We talked about this yesterday. Remember, this enhances scattering intensity at low angles, and that was a surprise. Right? And experimentally, when this appears, when you do dynamic light scattering, you see the slow mode. When you add salt, slow mode goes away. So does this one. So this detection of the large aggregate in a static light scattering or X-ray scattering, same as the large structure, which is manifest as very large radius of gyration of the aggregate, therefore diffusion coefficient is small. So we talked about the formation of the aggregate, again from counterions. Counterions are absorbing to the backbone, making dipoles. Those are dipoles get together to make quadrupoles, so they are more stable. Therefore, you make all these aggregates, and that's what this happens. Next one, please. Therefore, in summary, then, the ordinary exponential transition we see is essentially counterion physics. You would think, right? Why would I care about counterions? It's tiny. I'm interested in big molecules. That's what I'm interested in, right? But it turns out the small ions are hijacking the big polymer because there are too many of them, and you got to maintain electron neutrality. As a result of this, small molecules are these are like more like bees, hovering over a big elephant and carrying the elephant, right? So it's really amazing. But this is what has happened. Next one, please. Okay, so now I want to go to the other part of the question that I was raising in the beginning. How do molecules move in crowded environments? Already life is very exciting, and I think we understand what's going on at a low concentration limit. Now let's go to the high concentration limit. How because when you go and in here, in the nucleus of a mammalian cell, it's very crowded. Please pay attention to Goodell's book and various cartoons. We're extremely crowded. How do molecules move around? And how would a search engine, a protein will go and look for a particular part of the sequence of your mRNA or RNA or DNA? How is that going to happen? And in order to, I don't want to have a moving target. If this is moving around too much, right, it will be very difficult for a search engine to find a particular sequence. So it's extremely important to understand how these molecules are going to move around, diffuse around in an extremely complicated, crowded Coulomb soup. That's my question. And I cannot do that because it's too complicated. So I want to go to the next simplest situation. So what I want to do is I want to design a hydrogel, right? And I want to control the mesh size of the hydrogel. When I want to control the mesh size, then I want to trap DNA, for example, or any charged macromolecule. And I want to see how that's going to move around in this hydrogel. That is not exactly the same as either cytoplasm or nucleus, but I want to understand the physics of how these molecules might be moving around, how they are going to diffuse around. And let's go to the simplistic case. In a dilute solution where the mesh size, suppose I have a mesh size, a compartment like this room, is big enough compared to the molecule, right? This is my molecule, and my room, my mesh is like this auditorium, and this is going to diffuse around. There's lots of space here, just like a Stokes-Einstein, no problem. But now the room is small, or my polymer is too big, and I'm going to get it through that gate, through that entrance. Then it has to squeeze through. The polymer is big enough, it has to squeeze through. And then there will be an entropic barrier. When it squeezes through, number of conformations will be reduced, and that means the free energy will go up. So when you have a single barrier, there is an entropic barrier, and then what turns out is there will be e to the minus the free energy barrier coming from an entropic confinement, and that's all established in the literature. And then when you go to the ultimate limit, where the radius gyration polymer chain is very big, as a question that was raised before, and then the background is highly entangled, namely the mesh size is comparable to the monomer Kuhn length or monomer size. This is the beautiful work of Dijon and Edwards, the reptation model. Then the diffusion coefficient goes like 1 over n squared. So what we have diffusion here, this is like a ZIM. Here we have diffusion, which is smaller value, but it's diffusion because of room temperature. And then he's also diffusion, it's smaller than that diffusion coefficient, but much higher than this diffusion coefficient. Let's go to the next one, please. Okay, so the question then is, okay, I have diffusion here, Stokes-Einstein, or ZIM, and later on Rouse, because hydrodynamics is screened out. And then I have single entropic barrier, and this is my reptation limit. I was curious about this regime, because this was not addressed, right? What happens if the following situation arises? I have a molecule which is so big, lambda DNA, for example, is so big that it's going to occupy, in terms of mapping, it's going to occupy the whole building of this institute. All these rooms are connected. So one polymer chain will have parts of it in this room, another part in another room, another part in another room, but it's still a single molecule. Right? And this room is connected to the next room through one gate. Next room is connected to another room by another gate. So now I want to understand how this molecule is going to move, because that would be the intermediate case between this diffusion limit, this diffusion behavior, and this diffusion behavior. Namely, I want to look at radius gyration of the polymer to be so big compared to the size of typical rooms. And the size of the room is also big compared to the monomer size, because I want this polymer to be alive. I don't want this polymer to be dead, because I want this polymer to be functionally active inside the conditions that I'm imagining, cellular conditions. What happens? What happens if a number of entropic barriers act on a single chain? There's only one entropic barrier to the gate. There are many, many gates, so this chain has to get out. Okay, how would that happen? Then again, how would this transition from entropically controlled dynamics to entanglement take place from one barrier to another one? How does that happen? Okay, these are the kind of questions. So how does it diffuse? Next one, please. Here is a surprise. This is something we can do, right? We can do this. The way you do that is the following. Suppose I have some sort of a cavity here, and I put a certain number of monomers in here, and the radius of this cavity is R. I want to know the free energy of confinement. We could do path integral representation, do full calculation, or you can write a partial differential equation, diffusion equation, calculate with the right boundary condition. But intuitively, the free energy of confinement would be like this. This should be a function of radius gyration. N is molecular weight, number of monomers. It's a function of radius gyration to the radius of this confining area. Let me call this C, not to get confused. C is a mesh size or something like that. Now, this is then a function of n to the nu by C, because Rg goes like n to the nu. But we know that free energy is an extensive property. Therefore, this must scale like n, number of monomers. The only way this function, which has an argument of n to the nu over C, can be simply proportional to n, is that this is the power law. That this function is basically n to the nu by C power 1 over nu. Only then n per nu per 1 over nu will give me n. That means this free energy confinement will go like n divided by C to the 1 over nu. That's the only way it will depend upon measure. That's the confinement free energy. If it's a Gaussian chain, then this will go like n over C square. right? This is just a thinking process. You can work out the prefactors, all the numbers, 6 pi, everything you can work out very nicely. But the punchline is that the confinement free energy is going to go like number of monomers in that mesh divided by c squared. That's what, let's go to the next slide, please. That's what this equation says. Right, so I take this polymer chain, big one molecule, partition into different rooms, and in each room there is a M2, let's say M1 monomers. Let me take this one first one, M1 monomers. Then I calculate the confinement free energy. That goes like M1 divided by C squared. That's the formula that I wrote down there. That's coming from, then there is a tail here. That's because this logarithm is coming from there. There's a tie chain. So the number is different here. Instead of that, I have different number. This is the extra volume interactions we talked about in the last 2 lectures. The electrostatic interaction, all of them can be put in here. So I know the free energy of this polymer chain, which is spread out through these many, many meshes. Now I want to know how it's going to move. Next slide, please. Okay. I do know from other works in the laboratory, different other laboratories, the way a particular mesh is emptying the monomer there is by putting its head into the next chamber and torting, right? It makes the torch, and then the next one would follow. Then I calculated the free energy barrier of this. These are all exact calculations. When you do this, what happens is you have a barrier of a logarithm of this quantity, which depends on the mesh size and 1/96, right? But we calculated, I did, all right? That's a barrier. Normally, when you see logarithm, we don't pay attention, right? But I was fortunate enough to pay attention, right? Logarithm is very weak, that's what we think, but that's what we do. So, because that's the free energy, right? So the entropic free energy barrier. So the net entropic barrier essentially is that one. Then what I do is, what would be the time needed for the chain to the central mass to move a distance comparable to the own size? How long it's going to take for this to get out of the whole campus? How long that's going to take? And if you can calculate this, we are trained to do these things. When you do this, that's a 10 to the 19 times the time needed for a single monomer to go. Single monomer, maybe microsecond or something like that. So it's 10 to the 19. That's very long time. That's age of the universe, right? It's a very, very long time. So the time needed for the chain to move out would be infinitely long, according to this calculation. That means the chain is going to move. I told you right from the beginning. Einstein's law, diffusion coefficient is proportional to temperature. At finite temperature, room temperature, it cannot be 0. This calculation suggests that it is going to be 0. Right? And I told you that when somebody tells you like this, you have to check him out. Right? So let's make measurements. Let's go to the next one. Oh, one more thing before that. So now I told you that each monomer is alive. Next slide, please. It's alive in every compartment. So inside this compartment, there are M monomers, right? And there is a Rouse dynamics. Everything else is screened. And the rate for Rouse dynamics is 1/m squared. Confinement free energy is proportional to m, the way I told you there. And the probability that I'm going to have m monomers in one room is e to the minus Gibbs weight, Boltzmann weight, e to the minus m. Forget about those numbers. And then the light scattering measurements would be relaxation of the monomers in one chamber, and the probability of getting that number of monomer in that chamber integrated over all possible values. Then what you get is in light scattering, I should get e to the minus e to the 1/3, right? Not exponential law. Not only central mass doesn't move, but even the correlation function would not show the exponential law that I was telling you over there, right? Instead, I get this exponent, nothing to do with glasses. People see this glass is very, very low temperature, but that's not, this is water, room temperature, right? So non-diffusive hierarchical dynamics. Let's go and make the measurements. This next one slide, please. Oh, before that, I tell you. So this is the claim, right? The claim is there's a new state of polymer dynamics at intermediate confinement. That's what is bizarre about. Here we have diffusion, here we have diffusion, here we have diffusion, but somewhere in the middle, there's a death valley, right? This D is 0, according to this argument. But of course, that means, there's an apparent violation of Einstein's diffusion, non-zero temperature. It's an apparent violation. We are not violating Einstein's diffusion. We are really pushing the system into extreme metastability because the barrier is so large. So you're just trapping that, entropically trapping the polymer chain into a metastable state. It is all very well reviewed by experts in the world. Next one, please. Okay. So what we did was, I was very fortunate to have very talented postdocs in the laboratory. We made gels. We could tune the mesh size at will. We took different kind of polymers, lambda DNA, polypeptides, polylysine, many systems we have done. Variety of systems, we change the molecular weight so that we can meet the condition of RG being larger than mesh size, which is larger than monomer length, 96% water. Please, next one, please. Yeah. So hydrogel alone, that is the correlation function from light scattering, single exponential. And from there, you can get the modulus. That's okay. And polystyrene sulfonate in this particular case, solution, that's single exponential, that's okay. Now we take polystyrene sulfonate trapped in the gel. There is no single exponential. This is coming from the gel, very similar to this is coming from the gel. I can't see this. The first decay is from the gel. This is hierarchical dynamics, and that exponent is roughly 1/3. Just the way the analytical calculations show. Next slide, please. Also true for lambda DNA. Gel exponential, DNA solution exponential. When you put the DNA inside the gel, beta, that exponent is 1/3. Okay, next slide, please. We want to make sure we see it. Light scattering, you infer. So we labeled lambda DNA, a fluorescent label, and track it. And we track it. Okay, so nothing happened, it doesn't move. You run it for, until it bleaches, doesn't move. Do it again and again, doesn't move. Okay, then we want to know how much energy we have to give to make it move. What is holding it? How much is the barrier? So we put a little bit of electric field, DNA charge, put an electric field, nothing happened. We cranked up a little bit more, nothing happened. More, nothing happened. And when we hit the value 2.5 volt per centimeter, what happens is molecules took off. So for a given cross-link density, given mesh size, nothing happens. This velocity, nothing happens here up to that point. Beyond that point, there is mobility. So that value is a measure of the barrier. And then when you calculate the barrier, when you measure the barrier as a function of the mesh size, here is the diffuse regime, Rouse regime. This is the repetitive regime. In the middle, the barrier is very high, 120 kT. 120 kT, that's a very large barrier. That's equivalent to the 10 to the 19 time scale that I told you about. That's the reason why the DNA gets localized. It's not moving. Okay. This is very good news in terms of, okay, so this is a topologically frustrated dynamical state, entropically freezing. Let's go to the next one, please, very quickly. Okay, so this is very interesting for crowded environments. We always believe the molecules are diffusing, but not necessarily so. If it's really crowded, and the conditions are such that the molecules are very large, and the space in which they are moving are not small, they are big, but not big enough to hold the whole polymer chain, then this will happen. That means search engines, proteins, whatever they're looking for certain information on the molecule, they can do it more effectively instead of going around and around, chasing the molecule, which itself is moving around. So, and also, I did not talk about electrostatics in here. We can control the cargo, they charge the cargo right into this hydrogel media, and then you can release it at will. You can control back and forth. You can hold it and release it and back and forth. And you can also design, right, scaffolds to localize the DNA and also allow access to different things that you want to do with them. In addition to, of course, understanding how to harness extreme metastability for fundamental physics purposes. Okay. So next slide, please. This is probably my last slide. Okay, so what am I saying? This was our starting point 2 days ago. Polymers are Mother Nature's choice to express life. And as I repetitiously emphasized, it's all coming from conformational entropy. The temperature is fixed, right? Conformational entropy, the manifestation of conformational entropy with the collaboration of the counter ions and other small electrical ions, we are able to do lots of wonderful things, right? So therefore, given the title of our workshop, Soft and Living Matter, I believe it's a macromolecular in nature, and every information that I have shared with you so far is only baby steps. It's only the beginning, right? There are much more things that we have to do. Going to the next slide, please. As an example, if you take human cell, there are lots of organelles. They are membraneless organelles. And they are coming from intrinsically disordered proteins. They are more like a charged polymer, polyelectrolytes. It's not much more, right? Much more not like a folded proteins. They are intrinsically disordered proteins, which are implicated with many diseases, neurological diseases. One of the grand challenges today is to understand the confluence of all these things that we are talking about here: electrostatic, monopoles, dipoles, chain connectivity, hydrophobicity, water, counter ions, hydrodynamics, everything I have touched upon, which as a starting point. And I did not say anything about sequence because of interest of time. So these are the things which are conspiring together. They give me all these beautiful things. But when you look under the microscope, in addition to the formation of these ensembles, these aggregates, these very nice structures, when you look inside them, there are also very beautiful structures, self-organization of the fibrillation, things like that, right? And they are also implicated with neurological diseases. The structure and dynamics, they are very interesting. I think we, as a community, are in a very good shape to attack this grand challenge. There's a hierarchy of ordered disorder structures, the discrete functionalities, specific, all these things. We are, I think, on a very firm footing, fundamental footing to launch into these very interesting problems. So I want to show the last slide with a tremendous amount of thanks to you folks, right? For somebody on my side, the greatest reward in thinking about these problems is the younger generation to take over and attack problems of this type. And I hope some of the things that I shared would stimulate you to go and dig more and maybe perhaps contribute. Next slide, please. With that, I want to thank you for your attention. I'm completely open. Any questions? I finished early so that people can ask questions.
I was thinking you stimulated me. Now we're doing that again 26 years hence.
You too, please.
No.
No. You are very kind.
Okay, so then I have to say this. So Muthu ends saying that, he hopes the young people are stimulated. So I said I was 26 years ago when I was young. So I'm sure that you all will be too.
Well, that's very kind of you to say so, Ranjini. Life is good. And thinking is even better. If I can say that.
So you all are some questions.
There's a question over there in the back.
Loud enough. Yes. So, sir, the previous slide, 2 slides back, you are showing that micromolecules were not moving when you are observing through microscope. So it was completely not moving, or there are some fluctuation that we are not able to?
There are fluctuations. Thank you. It's not frozen. It's not a glass. It's not frozen, right? And they are fluctuating, but the center of mass does not move. The polymer is alive. It is not frozen into like a folded protein or a glassy-like structure, because look at the dynamic light scattering, the electric field correlation function showed that T to the 1/3 exponent, e to the minus T to the 1/3. That means there is a hierarchy of dynamics. It's not frozen.
So over a time, if we'll keep observing, it may move some distance.
No, we don't think so, because when you put the electric field, it doesn't go. We looked at, you are welcome to look at that particular reference in several places, right? PNA as reference. And we looked at the head angle distribution over a period of time, and we want to be careful about not bleaching it, by adjusting the concentration of the dye in it. We did very, very careful. The center of mass does not move.
So why I'm asking that question, because I've read these micromolecules sometimes get trapped, and that trap duration was widely distributed, like few millisecond to maybe hour.
We don't believe so, not in our system, because the mesh size is quite big, right? You can trap it in many ways. If you want to trap it, you can, right? For example, we did a case where the guest molecule is oppositely charged to the gel, right? There, you can trap it, and then you can release it by adding salt.
Okay.
So you could go to the athermal limit by releasing it, then it doesn't move, because this is the athermal limit. Like a DNA is negatively charged, and a polyacrylamide acrylate gel that's negatively charged. So the athermal system. So we chose that system, so therefore we don't have this trapping due to complexation. We didn't want to do that, right? So this phenomenon is robust. But if I make the mesh size bigger, it moves. If I make the mesh size tinier, then there's no entropic trap to hold it, because the room is tight and then it undergoes like reptation. So there's the sweet spot where the mesh size is satisfying that condition is the one that is localizing the polymer chain.
Thank you, sir.
Very good question. Thank you. Please.
And the previous question. So what if my polymer is more flexible?
Well, the theory actually goes for flexible chains. We did polystyrene sulfonate, lambda DNA, polystyrene sulfonate, same thing. It gets localized. I showed it, that's why I showed you two sets of data for PSS, polystyrene sulfonate. The X1 beta is 1/3. And then for double-stranded DNA, lambda DNA is again 1/3, universality class. And then we have labeled the polystyrene sulfonate, fluorescently labeled, and we see the same thing.
Which is playing the role?
The ratio of two ratios. Ratio of the radius gyration of the polymer guessed to the mesh size, and then mesh size to monomer length, right? If the mesh size is tiny, radius gyration is bigger, but the mesh size is tiny, then it will be in a reptation. Not in this particular case. That's going back to the previous question. Not in this particular case, because we are talking about single molecule measurements in here. Yeah. If it's really entanglement, then it will be more like a reptation, because the mesh size is tiny. In order for the entanglement to take place, the mesh size has to be, because of the distance between two segments is mesh size, right?
Thank you.
You're welcome.
Yeah. So in the dynamic light scattering experiments, where you have now you have a mesh also and a polymer also, so both of them can scatter light. So how do you identify which?
This is an excellent question, right? So yeah, so we have to make sure the refractive indices are different. Yeah, so that we can pick up gel and as well as the polymer separately. So we did the enormous number of experiments to separate it out individually. We know dN by dC and things like that, right? With the concentration gradient of refractive index, dN by dC. So these are different between acrylamide acrylate versus DNA, for example. So right. But it's a very tricky situation. You have to be extremely careful. So how do you separate it? Luckily for us, many other laboratories did light scattering on our gels. So we could benchmark our numbers cleanly with what is well known, right? So therefore, we are on a firm footing. So we are able to get, also fortunately, the decay rates are qualitatively different, quite different from each other. Therefore, we can easily pick it up.
So if both the components are in the Rayleigh scattering regime, then at any angle, both of them will.
Then they'll be different. No, that'll be difficult to do. In that case, it will be very difficult to do. In this particular case, it's okay. But there could be other situations where you may not be able to unscramble. Then you do neutron scattering. Label the guest molecule and follow it. We are doing spin-look experiments right now to look at local dynamics at this moment. There are ways to do it. We can deuterate only again. Or you deuterate the gel part and look at gel dynamics. Welcome to do these experiments. These are very nice experiments. Yeah.
For the discharge molecules, you said that if there, because of the counter ion, the diffusion is much faster than even the sodium or potassium ions. So
Comparable, less than that, but yeah.
So how it becomes? I mean, is there any physical insight? I mean,
No. See, this is like many situations, right? You have one relaxation rate, another relaxation rate. One is faster, higher, another one is slower. When you couple them, we are going to get two relaxation rates, right? Therefore, the slower one is going to be pushed to a higher rate. The higher one is going to be pushed to a lower rate. If I have free ions, counter ion alone solution, potassium alone, let's say chloride alone, without any polymer, it would have a higher rate, right? Higher relaxation rate. Right? Now, polymer will be a lower rate. When I have this coupling, I get into two new modes, right? The polymer mode will be pushed up. The other mode would be pushed down. But it so happens because of the coupling, the other mode, when it gets pushed down, it's not diffusive. Right? And when it's not diffusive, it's called a plasmon. There's a very famous work by Debye a long time ago called Debye mode on a plasma. Debye mode or plasmon mode. So we don't detect it in light scattering because the time scale, length scales are not there. But if I heard you right, I'm not saying this a faster mode, the diffusion coefficient higher than the diffusion coefficient of metallic ion. No, it's lower than metallic ion. It's about a factor of 4 or 5 smaller, but it's not higher. But I showed you the data. This has been tantalizing the community for a long time. We really could not understand why this is happening. Obviously, because it's bizarre, right? Many people repeated those things. We did not repeat. We did for other reasons, but we did not repeat them. Many laboratories repeated these things. They got it exactly. Well, thank you, folks.
Thanks once again.
Thank you.
So now we have a tea break.